This is National 5 Biology and topic 1.6, which is respiration. And I'm hoping that you recognise what is behind the word respiration there. That is the mitochondria. So this is where we're going to be in the cell, and probably all the way back in the beginning of S3, you'll have learned your parts of the cell, and we said that you probably learned that mitochondria is the site of respiration. So now we're actually getting round to talking about what respiration actually is, what occurs during that process. So you want to be able to explain what respiration is. Also describe the role of something called ATP during respiration. Describe what the energy produced during respiration is used for. And also, to begin with, we're going to look at, there's two types of respiration, aerobic and anaerobic, and we're going to look at aerobic to begin with. So respiration, put simply as a definition, and remember we're over mitochondria is involved, but also we've got the cytoplasm involved, which we'll get to soon. But respiration is defined as when cells release the chemical energy stored in glucose through a series of enzyme-controlled reactions. Okay, so glucose coming in to the cell as it's been broken. There's a little experiment we would do in class, and what you can do is you can get different types of food, and you can do an experiment and basically burn the food, and the longer that it burns for and the more heat it produces, that gives you an indication of how much energy or calories are contained within the food. So have a look at this little video that's on Twig coming up. We'll have a look at this, log in through Glow, search for measuring food energy, and that'll give you an idea of the different types of energy in food, and it'll give you a nice introduction to respiration as well. So respiration is turning the glucose, the energy in glucose, into another form of energy, and that the energy released during respiration is used to make a substance called ATP, and it acts as an energy store. So glucose itself, the body or cells cannot use that directly as an energy source, so what it has to do is turn it into ATP. ATP is short for adenosine triphosphate. That's just the chemistry of the compounds, you don't need to know that. Glucose, a form of energy that we can't use directly, but we can use ATP. So kind of imagine ATP being produced by cells. It's almost like charging up a battery, and then when we need that energy, we use the ATP for whatever purpose we need to. So you can have a think about various things that cells would do, that require energy. We've already covered quite a few of them. See if you can get any ideas from looking at these pictures. So first thing would be cell division or mitosis. Cells require energy for that, so they use their ATP store. That is a chain of amino acids. So chain of amino acids makes a protein. So protein synthesis requires energy. This you may well recognize as neurons. So to transmit nerve impulses, those electrical impulses along your nerves, that requires energy. And finally, I don't know if you'll be able to identify this, but this is a muscle cell, a smooth muscle cell. So in order to get the muscles to contract, that requires energy. Okay, I mean, there's a plethora of different things within the body that requires energy, but here's just a few examples. Now, we did say at the beginning there was two types of respiration. The first one is aerobic. So aerobic related to oxygen. And what happens during aerobic respiration is this. So there is the equation. So you've eaten, you've taken in sugar in the form of glucose. That glucose is combined with the oxygen that you're breathing in. and that is converted into carbon dioxide and water, and then the key thing is the energy. So this glucose being converted into energy, they can use carbon dioxide waste product which is breathed out. Okay, so aerobic, we have got oxygen. Now we talk about respiration occurring in the mitochondria, but actually the first stage of it takes place in the cytoplasm. So we're going to split aerobic respiration into two phases here. Okay, so we begin in the cytoplasm of the cell. And stage one actually doesn't require oxygen, and we call that anaerobic. So all that's happening during stage one is that glucose is converted into, here's a new term for you, pyruvate. Okay, glucose converted into two molecules of pyruvate, and as this occurs, you get 2 ATP produced. Okay, so stage one, oxygen is not used. We're in the cytoplasm, and you get only 2 ATP. What happens next is the pyruvate moves somewhere else in the cell. Where do you think that might be? Yes, so the pyruvate moves into the mitochondria, and that's where the next stage happens. So the next stage, oxygen does get involved, and what happens is 2 pyruvate combine with oxygen, and they are converted into the two end products, carbon dioxide and water. But the key thing here is you get many ATP produced. So you only get the 2 ATP produced in the cytoplasm when you don't have oxygen. When you have got oxygen and it's combining with pyruvate, you're getting many ATP made. So this stage, oxygen is used, you're in the mitochondria, and it produces many ATP. Okay. So here it is in summary. So first of all, you've got the first stage of respiration, which is anaerobic. Glucose gets converted into 2 pyruvate, 2 ATP produced. But then the key bit, when you move into the mitochondria, that is the aerobic stage. So it occurs in the mitochondria, it uses oxygen, and you get many ATP produced. So if you can recall all of this, I mean, I think if you can write out the flow chart, then that's the kind of key knowledge for this part. So a little video here. So hopefully, I think if you just Google BBC respiration, then you should get a little video on that, just to try and help you visualize it. Now here's a little task. So bit of paper, and we've discussed it. I'm hoping you've got some other resources as well that you've maybe had a look at, textbook, the printed notes and so on, maybe BBC Bite Size. But see if you can pause just now and see if you can do this, fill in the blanks. So stage one of respiration is anaerobic, which means there's no oxygen. So glucose converted into 2 pyruvate with the production of 2 ATP, and oxygen is not used, and it occurs in the cytoplasm. That's stage one. Next, try this then. So stage two, aerobic respiration, press pause. So we have pyruvate, the 2 pyruvate, which has been produced in stage one. So they go into the, well, I'll try not to give the game away for the answer down the bottom here. The 2 pyruvate combine with oxygen, and when that occurs, they are broken down with the production of many ATP to carbon dioxide and water. And clearly oxygen is used, there it is up there, and this occurs in the mitochondria. So the 2 pyruvate are produced in the cytoplasm, and they move into the mitochondria for stage two. And I want to have a wee break now, so go away, do something else, and then come back to this in a day or two, and we'll see what you've remembered. Okay, just a wee video again, as I like to kind of put these. I think it's really worthwhile. Don't just go through the PowerPoints and have a look at the videos as we go along. It'll help you get a better grasp of this stuff. So go and have a look in Twig and search for aerobic respiration. And to begin with, press pause and write down the answers to these. So just a sentence, or some of them it's just number 2 is a one-word answer, but write something for each of these. Okay, so first one, what is respiration? So respiration, a series of reactions controlled by enzymes in which glucose is turned into ATP. And this is a source of energy which cells can use. Where does respiration take place? I just said that was a one-word answer, and that's wrong, isn't it? Because it's not a one-word answer, because you've got the cytoplasm and the mitochondria. Number 3, what is the role of ATP in respiration? So ATP is produced during respiration as an energy store. Number 4, what are the possible uses of the energy produced during respiration? You could have a few here. So you've got mitosis, you've got protein synthesis, you've got nerve transmission, you've got muscle contraction. Okay, just revision of what you were doing before. And here's just to spell it out, there's a definition again. So respiration, when cells release the chemical energy stored in glucose through a series of enzyme-controlled reactions. That energy is used to make ATP, which is an energy store. And as I mentioned, there are various different uses for the energy stored in ATP, and there's just a few of them there. Okay, let's have a go at this again. If you haven't done this for a few days, then hopefully you've remembered it. So press pause and see if you can get the answers. So first stage of respiration, you have got glucose being broken down into 2-pyruvate. This produces just 2 ATP. Oxygen is not used, and this is occurring in the cytoplasm. That's your first stage. Press pause again, try this. So stage 2, pyruvate, the 2-pyruvate combine with oxygen. They're broken down, and this produces many ATP, and the end products are carbon dioxide and water. So oxygen is being used here, and the pyruvate has moved. It was produced in the cytoplasm, and it's moved into the mitochondria. Okay, so that's your stage 2. I just want to have a look at something called a respirometer. So a respirometer is something which gives you an idea of the rate of respiration. And remember your equation for respiration, glucose plus oxygen gives carbon dioxide plus water. So oxygen is being used up during this process. So if you can measure how much oxygen is being used up, that will give you an idea of the rate of respiration. And there is the equation there. Now, I'm just going to talk you through what we've got here. So in the middle, we have got a tube which has got coloured water in it, and a test tube which has got coloured water, and then it's got these delivery tubes that are in the water, and one of them is connected to this tube here, which has got germinating peas in it. So this is a living thing, so this should be carrying out respiration. And then over here, we've got the control. So this has got glass beads in it, which is not a living thing. So have a think about this. What's going to happen to the level of the coloured water in A? These peas are carrying out respiration. So what's going to happen is the peas are going to be carrying out respiration, so they're using up oxygen. So the oxygen in this tube and in this one are going to be used up. So as a result, oxygen is removed, so the coloured water is going to move up, okay, to replace the lost oxygen. It's going to fill the space, and you can see you've got your wee gradations here, so you can use that to come up with the rate of respiration. What about B? Level of coloured water in there? Well, in this one, that's the control, isn't it? That's not carrying out respiration, so nothing is going to happen there. So that should remain the same. Okay, so in B, the glass beads do not respire, so there's no change in coloured water level. Okay, that remains the same. Now there's a question you might have noticed. You've got cotton wool there, which is soaked in sodium hydroxide, and the sodium hydroxide combines with the carbon dioxide, so it's a neutralization reaction for your wee bit of chemistry. And why is it there though? Why would you want to absorb the carbon dioxide? Well, the reason you want to absorb the carbon dioxide is, remember that carbon dioxide is going to be produced by the germinating peas. So as a result of that, if you're trying to measure the rate of respiration using the rate of oxygen being used up, if the peas produce carbon dioxide, which is just left in the tube, it's going to kind of upset your results, isn't it? Because you're not going to, if the carbon dioxide is being added, then it will replace the oxygen as a gas that's being used up, so the coloured water would maybe not rise up the tube in the same way. So carbon dioxide produced by the peas must be absorbed, or it will affect the measurement of oxygen uptake. So that's why that's there, the sodium hydroxide. And just a wee validity question, experimental design. So have a think, what would you want to keep the same in each test tube in A and B? So pause and have a think about that, write down 2 or 3 things if you can. So things you'd want to keep the same. So you'd want to have the mass of peas the same as the mass of beads. Also the volume of sodium hydroxide. You can see they've got the water bath, so that's keeping the temperature the same. So a few different things you would want to basically keep the same in this test tube and this test tube. Okay, next up, this is just a little wee bit of problem solving. So 5 worms were placed in the respirometer and their oxygen uptake was measured. And first, so you can see that over time the oxygen uptake is increasing. So they're carrying out respiration. So a few things here. Present the data as a line graph. What was the oxygen uptake after 30 minutes? Predict the oxygen uptake after 70 minutes, and how could the results be made more reliable? So press pause and have a try at that. So I'll get onto the line graph in a little minute. The oxygen uptake after 30 minutes is just reading straight from the table, so it's 10 ml. Make sure you've got your ml in there, millilitres. Predict the oxygen uptake after 70 minutes. So what you're looking for is a sort of trend here. So you can see every 10 minutes it's gone up 4, 3, 5, 4, 3, 4, sorry. So you're going to expect it to go up between 3 and 5 again. So anything between 26 and 28 would be okay, I think, for that. How could the results be made more reliable? So to make results more reliable, you could repeat the whole investigation, so do it again another day with 5 different worms. You could use 10 worms, but it's just for B, just explaining a way in which you could repeat the experiment. For the line graph, just some common issues that people tend to have, mistakes that people made. Yeah, dodgy lines going through the middle of a cross, so you want to use a nice sharp pencil and ruler. Also joining to 0 when you shouldn't. Okay, you only join to 0 if 0 is in the table. You need to make sure you label the axes, so the X and Y units sometimes get forgotten when you're labeling axes, and also the 0 could be in the incorrect place. Okay, and just make sure you're going up evenly. Okay, 0 in the incorrect place, you've got your sort of a little right angle at the bottom of the graph here. It's easier to kind of put, if it's 0 for both axes, putting a 0 at the bottom. Some people have it kind of floating around here, and you have to make sure it's in exactly the right place to get the mark there. It's safer probably just to put in a 0 on the x-axis and a 0 on the y. Okay, have a break again and come back. Today's Tuesday, come back on Thursday or Friday. Okay. So on to the last part of this. So we're going to describe the stages of fermentation, and you're going to compare fermentation in animals, plants, and yeast. Fermentation is a word you will have heard of, and in terms of biology, or chemistry as well, I suppose, fermentation is the process which takes place when oxygen is absent. I've already talked about this actually, and glucose is only partially broken down. So in terms of the cell, we're only looking in the cytoplasm for this. Okay, so forget about the mitochondria for fermentation. So in animal cells, what happens is, we learned about this before, what does glucose get turned into? 2-pyruvate, and you get 2 ATP produced in the process. And then you might know this in terms of exercise. What does your body start to produce if you've been exercising for a long time? Causes a bit of pain. Lactate. Okay, or you're probably thinking lactic acid. But yeah, if there's a lack of oxygen, the 2-pyruvate gets turned into lactate. In plant and yeast cells, however, slightly different. You still get the first part, so the glucose is getting turned into 2-pyruvate and you produce 2 ATP. However, what happens is then the pyruvate is turned into ethanol and carbon dioxide. Okay, so a different process from us. And as I always like to say, if we had the same process as plants and yeast, then it would mean when you exercise, your cells would produce ethanol. So exercise would cause you to get drunk, which would make the Olympics more interesting, certainly. So differences between animal, plant, and yeast cells. So uses of the humans have uses for fermentation. You've got making bread rise, the carbon dioxide, and yeast does that. You might think, well, why is bread not alcoholic? Because it's got ethanol in it. Just the heat of the oven means it just burns off. Making cheese, yogurt, beer. This is all things that humans have used to take advantage of the fermentation process. So I want you to have this is a good little video on YouTube, History of Fermentation. The first time that humans drank alcohol, it must have been a situation where maybe people were starving and they saw some rotten food and they decided to eat it. It had fermented, and it had some effects that they maybe didn't expect. We're kind of evolved not to eat things that are rotted, but that's probably how we kind of started fermenting things. So have a wee look at this video. Now this is an experiment we would do in class, but we can talk about it and you can sort of predict what you think might happen. So what we've got is a delivery tube here, and it's going from one test tube which has got yeast, sugar and water in it to another which has got bicarbonate indicator. The other one, this is a control, so we've got the sugar but it's just with tap water and it goes into bicarbonate. Now bicarbonate indicator turns yellow if carbon dioxide is present. So what do you think? If you've got a test tube which has got yeast and sugar in it, is carbon dioxide going to be produced? So is the yeast going to make it turn yellow? And what's the smell going to be? If you've got no yeast, then what do you think is going to happen? So have a think about that. You might want to go back to the previous slide. So what is going to occur in the one where the yeast is with the sugar? you're going to start to have fermentation eventually. So to begin with, there's some oxygen up here, so you will have aerobic respiration. However, that will soon get used up, and once that gets used up, the yeast has to switch to fermentation. So it's going to start making ethanol and carbon dioxide. So we always enjoy this in the class because it's pretty stinking. You open this up and you get a kind of you get that kind of alcohol, slight alcohol smell, and also this turns yellow, so you've got carbon dioxide present. In the other one, and there's no sugar, so the yeast, well, it just smells of yeast. And this is something which people don't tend to enjoy cleaning out. But yeah, this just smells of yeast, and we don't have any carbon dioxide produced because fermentation is not occurring, so carbon dioxide is absent. Okay, here's just a couple little videos again. So have a look at the Twig film and fermentation, and do a Google search for BBC and processed cheese. Okay, let's do one of these just as revision of the whole topic. So there's 10 mistakes in here. So read through it, write down what word is a mistake, and write down what the correct word should be. Okay, press pause. Right, here we go. Respiration is a series of enzyme-controlled reactions which cells carry out to release the chemical energy stored in starch. It's not starch. That should be glucose. Its rate can be measured using a respirometer. The energy released from starch breakdown, that's wrong again, isn't it? So that should be glucose. The energy released from glucose breakdown is used to generate a substance called ADP, nope, ATP, which acts as an energy store. The energy transferred from, there's ADP again, can be used for cellular activities such as muscle cell contraction, cell division, protein synthesis, and transmission of nerve impulses. The more energy a cell requires, the more ribosomes it possesses. No, it's not ribosomes, is it? That's the lots of ATP are produced in mitochondria, so that should be there. So we've got 5 so far. In the cytoplasm, glucose is broken down into 2 molecules of pyruvate. That's okay. Releasing enough energy to yield 4 ATP. That's not okay. That should be 2. If oxygen is present, then aerobic respiration takes place within mitochondria, with each pyruvate being broken down to carbon monoxide, water, and many ATP molecules. That's a mistake. It's not carbon monoxide, it's carbon dioxide. In the absence of oxygen, fermentation occurs in the vacuole. Where's that come from? That is a load of rubbish. That should be cytoplasm. In animal cells, the pyruvate molecules are converted to amylase, no, lactate or lactic acid, whereas in plant and yeast cells, they are converted to carbon dioxide and water. That's not right either. It should be carbon dioxide and ethanol. Okay, so there's our 10 mistakes. I think this table is a nice little summary of the differences between aerobic respiration and fermentation. So I'd like you to draw this out and try and fill in the blanks. So press pause and then we'll go through it. So first of all, aerobic respiration, oxygen is required. Fermentation, oxygen is not required. Aerobic respiration occurs in the mitochondria, whereas fermentation, no oxygen, that's occurring in the cytoplasm. For aerobic respiration, you're going to get many molecules of ATP. For fermentation, you're only going to get the 2. End products, for aerobic respiration, carbon dioxide and water produced, whereas fermentation, lactate. So this is animal cells. And then aerobic respiration, the end products in plant and yeast cells is carbon dioxide and water, whereas in fermentation, it is carbon dioxide and ethanol. Okay, so just a comparison of the 2 processes that we've been learning about. This is possibly the final slide. I'm not entirely sure, but let's see. This again, just a wee exam question. This is from 2016. A muscle cell will carry out fermentation when oxygen is not available. describe the fermentation pathway in muscle cells. Okay, so you could do a drawing for this, the wee flowchart if you like, or a description. Let's see if you can get the 3 marks. So press pause and have a go at that. Okay, now the first thing about this that you have to be a bit careful about is it says describe the fermentation pathway in muscle cells. So plants have muscles, the yeast have muscles? No. So what they're looking for basically is the fermentation pathway in animals. So here's your marks. First of all, glucose is turned into pyruvate. Second of all, pyruvate is turned to lactic acid or lactate, and you get ATP produced, 2 ATP produced. And you can see here arrows are acceptable in place of an explanation. So even if you did that very simple wee flowchart, glucose convert to pyruvate with 2 ATP produced, and then an arrow going from pyruvate to lactate, then you'd get your 3 marks there.